Hi, I'm Stacie Calad-Thomson. I'm the Chief Strategy Officer and Head of Drug Discovery at BioSymetrics. High attrition and clinical failures are the biggest problem in drug discovery, and most of this is due to incomplete understanding of biology. But there's a huge opportunity to bridge this gap with patient data. However, I've seen how clinical data and patient results are siloed from the discovery process, and how learnings aren't often shared between or even within those organizations. Additionally, that data is complex and noisy, and therefore a difficult starting place to initiate drug discovery programs. Instead, we typically distill that complexity down into single protein target-based approaches that are sequential and linear. But if that target hypothesis was incomplete or not widely representative of diverse patient biology, we risk spending a lot of time and money on that process before discovering a lack of translation in the clinic. Our mission at BioSymetrics is to translate data into new discoveries. Our approach is driven by phenomics and machine learning, where we integrate clinical data and experimental learnings to better navigate disease biology and advance precision medicines. The foundation of our approach is our Elean platform, which enables our phenomics approach and allows us to navigate human disease biology with a goal of advancing new drug discovery programs. Our 4 modules work together to integrate and associate clinical insights, genomics, and phenotypic data to better understand the complexity of pathways and targets needed to best modulate a given disease or phenotype with a small molecule. The Phenograph is at the center of this platform. It's our knowledge graph that integrates and associates human genes and phenotypes to those of model systems and through to small molecules that impact phenotypes. Through this linkage map, we can better navigate disease biology, and we can query it at every stage of drug discovery to better guide us in the right experiments we need to run to connect data and improve predictions. The BioSymetrics secret sauce can be boiled down to 3 things. The first is our unique access to data, both clinical patient data and experimental data. The second is Contingent AI and our machine learning models and infrastructure. The third is human disease relevant experimental models, both cellular and in vivo, that help us validate predictions as well as generate new training data sets. Our data access is one of our biggest differentiators. We have partnerships with some of the nation's largest hospital networks and access to over 13 million patient records and counting, which amounts to over 100 million longitudinal patient years. Through our partnerships, we've systematically and repeatedly worked with partners to ingest, clean, and curate these massive data sets. We're combining it with other data, public, private, and experimentally generated, to create a massive linkage of biomedical data from which to initiate drug discovery programs. Some of this data is phenotypic data, including massive in vivo data sets that we generate with our partners like Brigham and Women's Hospital, Harvard Medical School, and University of Toronto. We're also leveraging open data sets from some of these sources on the bottom and continuing to generate new drug discovery associations ourselves. The way that we're able to use that vast data is with Contingent AI, our patent pending software that enables us to denoise and integrate diverse data. Contingent AI is an automated framework that expands the model building process, resulting in less time spent in data processing, easier data integration, and more accurate results. This iterative pre-processing approach reduces bias and helps us achieve better, faster models from the data. AI predictions, and for that matter, exploratory drug candidates, are only as good as their experimental validation. So the third component of our Elean platform is our human relevant phenotypic models of disease. We use zebrafish, cellular, and other models to generate training data and validate predictions. Zebrafish have been a major focus of our platform thus far, since they're transparent vertebrates that offer a high throughput screening model system with the benefit of whole organism in vivo biology that can be genetically modified with CRISPR. Our computer vision platform is enabling the scaling of our experimentation by helping us to rapidly incorporate new high quality data into our active learning process. Our partnerships have enabled us to validate our Elean platform at every stage of drug discovery and drive forward drug discovery programs. In the last year, we've worked with Northwell Health, Jansen, and Humanigen to provide clinical insights for COVID-19 projects. With Merck and Pfizer, we've undertaken data-driven projects to identify the mechanism of action in phenotypic drug programs. And in our ongoing relationship with Sema4, we've successfully analyzed patient records to provide clinical insights and then progress those insights into drug discovery programs. In our atherosclerosis program with them, we've prioritized genes and progressed the program through to a phenotype and hit screening. This model of data to discovery is what we're now replicating with plans for 10 new programs initiating in 2022. Our Elean modules can work together in multiple directions, but we often start a process from patient genomics and electronic health records. Our team of data scientists is able to curate this data to provide actionable clinical insights and next steps based off of patient phenogroup clusters with identified biomarkers and genes associated with a disease. In our next module, gene disease prioritization, we've trained hundreds of machine learning models to predict gene disease associations and based on their probability to impact a phenotype of interest. Starting from a long list of associated genes from our patient phenogroups, our models help us predict the top associations, thereby narrowing the experimental space where we can come in and use CRISPR models to validate our gene predictions and observe phenotypes. Once we've got a genetically associated phenotypic assay, we can initiate our third module here, which is small molecule screening. Our machine learning modules can reduce chemical space and prioritize screening libraries based on predicted structure phenotype associations. And in our final module here, we can uniquely address one of the main issues with phenotypic screening. We can take results of hits and non-hits from a phenotypic screen, and our mechanism of action engine can uncover drug targets, pathways, and predict expanded hits and chemical space computationally. By using our 4 Elean modules together, we're able to tackle complex polypharmacology and initiate drug discovery programs with a better understanding of disease biology drivers. Our partners have shared millions of patient records with us in exchange for deep analytics and insights that can help them derive value from this data to better help patients. In our work with Jansen, a J&J company, we embarked on a program last year that ultimately helped them stratify patients for the J&J vaccine clinical trials. We started that work with data from over 20,000 patients tested for COVID at Mount Sinai in New York that were collected during the first wave of COVID-19 in 2020. Using machine learning, we were able to identify distinct patient phenogroups and corresponding biomarkers, and then predict the onset, severity, and disease progression of COVID-19 in those different groups. Additionally, we've carried out work with Northwell Health on predicting acute respiratory distress syndrome amongst both COVID and non-COVID populations, which is helping them differentiate patients requiring mechanical ventilation in the ICU. Going forward, we aim to use this process of phenogrouping patients and identifying biomarkers and gene associations as starting points for drug discovery programs. Sema4 has been a great partner to us that we've worked with on several programs thus far, and we'll soon be embarking on more programs to come. In this example, I'd like to highlight our atherosclerosis program with them. Starting with data that they had access to from Mount Sinai on over 10,000 patients, we were able to pull out gene associations. And using machine learning, we prioritized a list of 20 genes that we predicted to be associated with atherosclerosis phenotypes. We then used CRISPR and zebrafish to knock out those genes and observe the resulting experimental phenotypes, and found several novel genes that displayed disrupted vasculature. On the right, in the top image on the bottom image is one of those genes. In the top image, we've got the wild type with, as you can see, complete intact vasculature. And on the bottom is an example of one of our genes of interest that was CRISPR'd, and you can notice a difference in and a disruption in the vasculature there. We're now undertaking additional cellular, phenotypic, and mechanistic studies, as well as embarking on small molecule screening to identify probe compounds and further validate the potential for these genes to be novel disease targets. Our team. I joined BioSymetrics full-time several months ago, and the incredible team here was one of my biggest reasons for joining. Anthony and Matt have worked together in previous successful startups. Gabe has an extensive academic research career with expertise in both computational and experimental biology work, including working with Callum McRae, who's been incredibly involved as an advisor at BioSymetrics from day one. We have a strong scientific advisory board and a growing team of data scientists, experimental scientists, and drug hunters, with hubs in Toronto and Boston, as well as the ability to tap into a remote workforce and access the best talent anywhere. To recap, BioSymetrics is a phenomics-driven seed-stage drug discovery company with plans to initiate 10 new programs in 2022. We're seeking pharma, clinical, and genomics partners to join us on this journey, and we're also seeking bio and tech investors for our next round. You can email me here or connect through the Biotech Showcase partnering platform. I look forward to hearing from you.